What is the purpose of interphase? The purpose of interphase is so that it just allows the cell to function as a normal cell and develop so it can get to the point where it can start mitosis.
What are three parts of interphase and there are three parts of interphase: G1, S phase, and G2. And the G1 is the growth one, in which the cell creates organelles and begins metabolism. The S phase is where the DNA synthesis, in which the chromosomes and the cells are copied. And the G2 is the growth 2, in which the cell grows in preparation for the cell division.
What is the purpose of prophase? During the prophase, the nuclei in the nucleus disappear. So this cell right here would all disappear. So you'd only see these. And then chromatin fibers become tightly coiled. So this becomes tightly coiled, and this becomes tightly coiled. And the chromatin fibers become the chromosomes lined up in pairs and are joined in the centrometer. In the cytoplasm, the spindle fibers begin to form and are made of microtubes. Tubules. The centromes move away from each other and are propelled by lengthening microtubes or spindle fibers between them.
What is the purpose of metaphase? The centromes are at the opposite ends of the cell poles. The chromosomes are lined up at the metaphase plate, an imaginary line equidistant between the two poles. The centromes of the chromosomes are aligned with one another. Their chromatids of each chromosomes are attached to the chromatube, which are from the spindle.
The purpose of anaphase. The purpose of anaphase is that the paired chromosomes separate. The paired chromosomes separate, and they move along the microtubes towards the opposite poles of the cells. And the two poles of the chromosome, they each now have a complete set of exactly the same chromosomes. The purpose of telophase and cytokinesis. In telophase is to reform the nuclear envelope, like this. It'll start the reforming of the nuclear envelope, and which starts the formation of new cells. And then in cytokinesis, it completes the nuclear envelope like this. So it's basically a new fully functional cell of its own. It's two new functional cells. And
What is the purpose of mitosis? It is to create two new cells out of one old one.
Purpose. What is happening to the DNA during this during mitosis? Well, what's happening to it is that in order to become two new functional cells, it's going to have two new functional strands of DNA. So this one strand of DNA is going to have to become split and be copied into becoming two strands of DNA.
Are the cells identical or different? Why? The cells are completely different because they...
Oh my, you just said different. Sorry.
Are the new cells identical or different? Why? They are identical because they need to create or do the same task that the old cell did.
Where does mitosis occur and at what rates? Mitosis is the duplication of the nucleus, so therefore it takes place within the nucleus, and it occurs in most mammals. It takes the rate of 24 hours. So that's pretty quick, and this is proven by 90% of cells are in their interphase, and so that means that if it took longer and if it was a slow process, then most of their cells would be trying to split, and it would be the other way around, where 90% were in the process of mitosis, and the 10% would just be in interphase.
How does cancer relate to mitosis? In order for cancer to spread, it needs to duplicate, so it uses mitosis. It also spreads faster because of how fast it does the mitosis process.
If it makes a mistake during the process of mitosis.
I don't know.
If it makes a mistake, then one may turn out normal, but then there might be a mutation in the other one. Like this is normal, where there's two green, one from the mom, one from the dad, and two purple, one from the mom, one from the dad. And here there's purple, two purple from the mom and the dad, but the green one accidentally transmuted a yellow chromosome which made it, which is a mutation, which means this is a dysfunctional cell which isn't able to carry out. This is the centriole, and these things that grab onto that actually grab on are the spindle fibers.
Here's the cell, and here is the nucleus, and in the nucleus is the chromosome, and in the chromosome holds the DNA.
The nucleus is the nuclear membrane.
It becomes two. It becomes sister.
The cell is the cell membrane, and within and once it splits, it creates the two daughter cells. Like once it splits from this, it creates the two daughter cells, which are these. And these are the sister chromosomes, but the cells. But the original cell is called the parent cell, which is the cell before it splits into the two sister cells.